Hello everyone, my name is Eduardo Lamas, and in this video we are going to talk about inflammation. There are three stages of inflammation. In stage 1, there is vasodilation and increased vascular permeability. Two of the main cell types found in stage 1 include macrophages and dendritic cells. The activation of TLRs will lead to the secretion of cytokines and chemokines, such as those pro-inflammatory molecules that we've seen in previous videos. A third cell type that is seen in stage 1 of inflammation is mast cells, which secrete histamine that can lead to local vasodilation. Additionally, we have activation of the complement system. We also have the activation of platelets, which participate in coagulation, and of the kinin, prostaglandin, and leukotriene pathways, which can lead to different effects such as vasodilation or vasoconstriction. In addition, these pathways can lead to increased permeability, resulting in edema. Stage 2 is characterized by polymorphonuclear cells and macrophage mobilization. Leukocytosis-inducing factor will increase neutrophil production. Stasis is also seen in stage 2. Stasis is basically the slowing down of blood flow. In this case, an inflammatory stimulus causes junctions between cells to open up wider than normal, which allows excess fluid to get out of the vessel. After fluid leaves the vessel, the blood gets thicker because of the large protein particles and cells which stay inside the vessel. These characteristics are the ones that lead to a slowing of the blood flow. There's also margination, in which white blood cells line up along the peripheral edges of the vessel. There is also diapedesis, or the movement of leukocytes into the tissue from the blood vessels. We also see chemotaxis of white blood cells, such as neutrophils, which are among the first cells to arrive, and monocytes, which arrive later. Stage 3 of inflammation is characterized by tissue repair. There is phagocytosis of pathogens by polymorphonuclear cells and macrophages. There may also be the formation of pus, which is the debris of dead cells such as phagocytes and other white blood cells, damaged tissue, and even microbes. If pus is too much for effective removal by phagocytes, an abscess may develop. Finally, there is tissue regrowth and repair of damage or scar formation. Inflammation causes increased plasma filtrate to seep into tissue and bring some immune proteins with it, leading to edema. This process helps to dilute harmful substances, increases the supply of oxygen and nutrients needed for metabolism, inflammation, and repair, increases blood flow that leads to increased local temperature and cellular metabolism, increased capillary permeability and phagocytic migration to the injured tissue, and it allows the entry of clotting proteins, which reduces the spread of microbes. Tissue injury leads to a release of different chemical mediators, such as histamine, prostaglandins, complement, and kinin, and to a release of leukocytosis-inducing factor. Chemical mediators and leukocytosis-inducing factor will initiate two separate pathways that will contribute to the symptoms of inflammation and to healing of the injured tissue. The release of chemical mediators has four main effects. First, there is arteriolar vasodilation. This causes an increase in blood flow to the injured tissue, also known as local hyperemia. The increase in blood flow will produce redness. The increase in blood flow will also produce an increase in heat or temperature, which leads to an increase in the metabolic rate of cells. In addition, local hyperemia will increase oxygen and nutrient delivery to the affected tissue. The second effect of the chemical mediators is the generation of pain. The third effect is the increase in capillary permeability. This leads to a leaking of fluid, which causes formation of an exudate, or protein-rich fluid, into tissue spaces. Leaking of fluid slows down blood flow, and this allows leukocytes to cling to capillary walls, also called margination. The exudate is also responsible for causing pain and swelling, both of which can temporarily limit joint movement. It is important to note that swelling adds up to the pain caused by the exudates. Clotting factors can also be leaked and will form blood clots to enclose the affected area and prevent the injury from expanding to adjacent tissues. This process involves the deposition of transient fibrin patches, which constitute a scaffold for repair. Finally, there will be chemotaxis of white blood cells, such as neutrophils, monocytes, and lymphocytes, to the injured tissue. On the other hand, the release of leukocytosis-inducing factor increases the number of leukocytes in the blood, known as leukocytosis. This allows for migration of white blood cells to the injured area. Then, there will be leukocyte binding to the capillary walls, also known as margination. This is followed by diapetesis, or the passing of leukocytes through the capillary wall into the tissue. Then, there will be phagocytosis of dead tissue and pathogens by neutrophils in the short term and macrophages in the long term, with possible formation of pus. The end result will be the clearance of debris from the affected tissue. After watching this video, please rate it by using the thumb up or down option below. Thank you.